All right, everybody, this is my attempt to start a online review for AP Bio Unit 1. If you remember back when we started the year, this probably took us several weeks to get done, and we're going to do it in one video right here. So, just a couple things. I'll probably make a few mistakes in this. That's unavoidable. Video teaching is a little bit harder to clean up those mistakes. If you find one, please point it out to me in the comments. Ask lots of questions in the comments for this assignment, okay? For this assignment. Please don't email them to me. Ask them in the discussion tools available for this video assignment. Just something to think about. The questions we're probably going to see are about content and skill. There's a certain amount of skills that are in the course description. I'm going to give you a link to those below. Just make sure you read over those skills, and I'll talk about those skills as we go. And lastly, your mental health right now, especially in this crazy time, is way more important than exam prep. So please just take care of yourself. Take care of yourself, take care of your health, take care of your family. I encourage you to use all these awesome electronic tools we have to do some sort of meetups of your own to study. If you're setting something like that up on a school-approved platform, you can invite me and I'll try to drop into it. You can make your own meets, and or you can ask me to set one of those up, and if I can make it, I will. All right, our first topic is structure of water and hydrogen bonding, and our job here is going to be to explain how the properties of water that result from its polarity and hydrogen bonding affect its biological function. So let's take a look at that together. Okay, here we go with some water. This should be easy stuff. You've got your oxygen and two hydrogens. Those bond covalently, so electrons are shared equally between oxygen and hydrogen, but that still leaves a little bit of a negative and positive charge, a partial negative and positive charge on the oxygen and hydrogen ends, and these cause weak hydrogen bonds shown with the dotted line. So water develops some properties due to these bonds we're talking about. All right, these different bonds we're looking at, especially those hydrogen bonds, give water some of the properties we looked at this year. One of those is cohesion. When we put the water on top of a penny, we saw that the water could sort of build up and stack up into a pile, and that's because hydrogen is holding the droplets together. When you mix things with the water, it interrupts those hydrogen bonds, and that doesn't work as well anymore. All right, here's just a picture reviewing that. Cohesion is water-water bonding, and adhesion is water in something else that has some polarity. Now, water can't hold on to everything. Whatever it's holding on to has to have some polarity. So if you think water might fly right off of a piece of glass that doesn't have hardly any polarity, but it might stick really well to another substance that has polarity. So adhesion is different, cohesion, water-water. So your example of this is water movement in plant stems. Water is attracted to the cellulose in the xylem tissue, and that helps it adhere and climb up through the xylem, pulling other water molecules behind it due to cohesion. So cohesion pulls the other water up behind it, and those water, that collection of water is not going to pop apart and leave an air bubble in there because of cohesion. It will pull more water up behind it, like everyone holding hands. All right, here's a little chart that shows properties of water. We talked about hydrogen bonding. Another one is water's thermal conductivity. Water expands when it freezes, so that makes ice float, which is a really interesting property biologically. Without that, bodies of water might freeze up. Water can hold a lot of heat. It's very good at absorbing a lot of heat before it increases in temperature. If you've ever had to make macaroni and cheese, you know how impatient you can get. All right, there's cohesion, adhesion. We already talked about those briefly. Surface tension is due to cohesion, so certain bugs and things can walk across water. Heat of fusion is how much it takes to change the state. It takes quite a bit of energy to do that. Polarity, the oxygens are slightly electronegative and the hydrogens are positive. Heat of vaporization is just like heat of fusion. It takes a specific quantity of heat to cause water to be vaporized or boiled. And because it has that polarity, water is a very good solvent and it can dissolve many, many different chemical substances biologically. Okay, that's the end of water, and we're moving on to elements of life. We're going to look at the composition of some different macromolecules that we've had hanging up in the class all year round on that bright colored fluorescent paper. And these are all required by living organisms. So let's take a look at the structures of these and what's important for us. All right, this section is a little bit short. We're going to be looking at some things we've looked at all year round. And after being in the class for a year, hopefully you remember things like carbohydrates, lipids, proteins, and nucleic acids. So let's talk about those real briefly and then we'll get going. This is a review, so if you need more, you're going to have to go back and look at your materials or the textbook or ask for help. All right, here's a little graphic that has everything summarized for you. Let me zoom into some areas and we'll talk about it. All right, macromolecules are big. Macro means big, and they are organic compounds. That's C-pounds, they're abbreviated. And they all contain some sort of carbon, so that's everything on the chart. All right, let's start with our carbohydrates. In each one of these, we're going to have monomers, the units of the chain usually that make up these macromolecules. Okay, so we're going to go from monomers to polymers. Okay, look at the top of your screen there. Make sure you know the elements in carbohydrates: carbon, hydrogen, oxygen, in a 1-to-1 ratio. Then you've got mono, di, and polysaccharides. That should be pretty easy for you. Glucose, everybody should have that chemical formula memorized. There's a glucose ring. Most of these are going to end in ose, and they include things like starches, cellulose, glycogen, and glucose. There's lots of different sugars we looked at this year. Those are the common ones. I'm not going to go over all of them right now. Okay? Carbon, hydrogen, oxygen, 1-to-1 ratio. Okay, next we have lipids. Lipids are going to give us fats, oils, and waxes. Okay, the monomer for a lipid is a triglyceride, which is going to be 3 fatty acids and a glycerol holding those together. Now, fats come in 2 different varieties we looked at. The first is saturated. In a saturated fat, each carbon is fully loaded with hydrogens. There are no double bonds. In a saturated fat, there's just no more room to put hydrogen on. Now, look at the unsaturated fat. Right in the middle, you see a carbon with a double bond, and there's only 1 hydrogen coming off of each of those 2 carbons that are part of that double bond. So an unsaturated fat has more room to add hydrogen if those bonds were not doubled. So if those bonds became a single bond, you could load more hydrogen on there. Okay, let's move over to proteins. Proteins are what enzymes are made of proteins, and almost all of the cell structure is made out of proteins, and proteins do a lot of different things. They're incredibly versatile. For proteins, you're looking at carbon, hydrogen, oxygen, and nitrogen. This group of macromolecules contains nitrogen, and some contain sulfur. The monomer is an amino acid, and those are going to be bonded together by peptide bonds. And we looked at how those are formed. The ends with the two hydrogen and the oxygen and hydroxide group, when those form together, we get water out. So that's going to be a synthesis reaction that's going to create chains of amino acids that will fold up into your proteins. All right, our next macromolecule are nucleic acids. And nucleic acids contain carbon, hydrogen, oxygen, nitrogen, and phosphorus. We have DNA and RNA, and we look at the different bases over there with the A, G, C, T for DNA. And for RNA, it's going to be A, G, C, U. So here we have the sugar rings, the nitrogenous base, which has the nitrogen in it. You can see the base there has a lot of nitrogen in its ring. And then we have the phosphate backbone. All right, that kind of concludes section 1.2. We're going to go ahead and start heading into 1.3. All right, so in this section, you're going to describe the properties of the individual monomers and the kinds of bonds, which I started getting into in 1.2. But right now, we want to look at those monomers, their bonds that make up these macromolecules. Okay, I'm sitting at my kitchen table, and I just pulled these off of the little Lazy Susan rack we use. And these are going to be my monomers. They're going to be individual units. Now, if I chemically bond a bunch of monomers together, those join a chain and become a polymer. So we're going to talk about the kind of bonds that hold these together and what are the individual units of these polymers called. So the monomers for proteins are going to be a lot different than the monomers for carbohydrates. Let's take a look. Okay, let's just look at these different polymers, which we did in the previous section, and their individual monomers. Your carbohydrates are going to be rings called saccharides, which means sweet. Your proteins are going to be individual amino acids and their R groups. Your lipids will be fatty acids. That's a little bit of an oversimplification, but we're not going to spend time on that. And your nucleic acids will be little nucleotides that have 3 parts to them. All right, let's take a look about how these monomers and polymers get put together and taken apart. In dehydration synthesis, you take monomers and put them together into a polymer, and water is removed, okay? So water comes out. That's why it's dehydration. In hydrolysis, you add water, and the water breaks the covalent bonds between the monomers in the polymer. And the diagram kind of shows that really well. Just pay attention to which way the water's going and which way you're going, either from polymer to monomer or the other way around. Just remember what the word means. Hydro means water and lyse means cut. And so we are literally water cutting. All right, that wraps up 1.3. 1.4 is properties of biological molecules. What we're going to do is dig into each one of the types a little bit deeper and take a nice close look at each one. All right, I will post this really well done summary sheet somewhere on the webpage. All right, carbohydrates are where we're going to start. And carbohydrates are sugars and polymers of sugars. So they're made out of these individual sugar rings. And they can do a bunch of different jobs. They can store energy, like in starch or glycogen, and they can provide structure, like cellulose. All right, we start with monosaccharides that are going to usually roll into rings, which you can kind of see down here toward the bottom of the page. If I can zoom in, there's your ring right there. That's the limit of my zoom in, but it's right in the middle of the page pretty much. Now remember, these are always going to be in that 1:1 ratio. And in this picture right here, it's shown as X2XX. So that's going to be a good way to tell if something's a sugar. All right, if you hook two of these sugar rings or saccharide rings together, you get a disaccharide. And those form what are called glycosidic linkages. Well, let's talk about those. Forming those is a dehydration reaction. And so when you dehydrate something, you take the water out. Polysaccharides, that chain is just going to get longer than two. So 3 or more are going to give you a polysaccharide. And just remember, if you have 2 rings, you make 1 water because there's only 1 link. If you have 3 rings, you make 2 waters. And if you have 4 rings, you make 3 waters, and so on. So watch for that. I know there was some confusion on that when we learned this earlier in the year. Do me a favor. I will put this image of really well sketched out notes into a link below this video or somewhere around this video. Take a look at this and maybe print this out to have nearby when you do your test. All right, let's move on to lipids. In front of me, I've got 2 different types of lipids. I've got an animal lipid here. I've got a plant lipid right here. It's my olive oil. All right, let's talk about what makes these different. All right, I will throw this into your the page that I put this video on. So lipids are made up of fatty acids, usually attached to a glycerol backbone, okay? And in biology, they're gonna show up as fats, oils, and waxes. So you think of plant oils, animal fats, and some, earwax is a lipid, and of course insects like bees use waxes in different ways. The other thing we see them forming is steroids like testosterone and estrogen, okay? They do make up 2 different types, which we can see down here at the bottom. You've got your saturated. They are solid at room temperature, and that'd be things like the butter I showed you. So every single carbon is holding as many hydrogens as it can. And then you've got your unsaturated, where there's going to be some sort of double bond that kink the molecule, and those form liquids at room temperature. Take a look at this summary and maybe have it nearby when you take your test. One of the big things you want to remember with lipids is that they are non-polar and they do not dissolve in water. And there's one exception to that, and that's phospholipids, and that phospholipid head is actually hydrophilic and is attracted to water. Okay, we're going to switch over to proteins now. So proteins, and again, this diagram will be over on the web page. Proteins have several functions. They give structure. Okay, they are used for signaling. So when we get into cell signaling, they'll make up things like neurotransmitters. They are catalysts, and so that's what we're going to be calling enzymes later on. And they do a lot more than that, but those are the big functions of proteins. The monomers of proteins are amino acids, which have an R group on them, and they're made of different functional groups that we're going to talk about, and they have lots and lots of different chemical properties. Now I've zoomed in here to the thing that all amino acids have in common. The R can be many, many different things, but each amino acid has an amine group and a carboxyl group on each end, and they are directional, so they're going to hook up with each other through dehydration reactions, and each carboxyl will bond with the amine side, and the carboxyl will lose the OH, and an amine will lose 1 hydrogen, and those will form water. So that's how we get amino acids to form polymers from monomers. Now, the different R groups can be really have huge amounts of variation in them. All right, we talked a little bit about R groups. You do not have to know the names of any of the R groups, but you should know they have different chemical properties. They can make R groups. They can make sulfur bridges. They can be hydrophobic. They can be hydrophilic. They can be ionic. And the way these R groups behave is going to determine how the protein folds and how it interacts with different chemical structures. Remember, these are molecular machines that are designed to interact with a vast number of different chemicals in organisms. So they are universal machines. So it kind of makes sense that they're going to have all these different functional R groups because they have so many different jobs. All right, our next molecule we're going to talk about and our final one are nucleic acids, okay? Nucleic acids are very large biomolecules and they are in cell nuclei. These are polymers that are linked in a chain of nucleotides, which are what we're going to look at right now. And of course, functionally, these are for information storage. All right, a nucleotide is made up of three elements here. You have a pentose ring of sugar, and pent just means 5 here, and that's the middle gray one in our diagram. Number 2, you have a phosphate group, which is this purple circle on the left, okay? And then you have the nitrogenous base, and that's either going to be your A, G, C, or T. These link together in condensation reactions, forming what are called polynucleotides, which make up one of the rings of the double strand of DNA. All right, moving on to 5.1. And in this section, you're going to have to explain how changes in subunits of a polymer might lead to changes in structure or function of a macromolecule. Okay, I've talked about these quite a bit already throughout the lecture. Just know that when sugars, for carbohydrates, when sugars make these chains, that it is a dehydration reaction and water is going to come out to form those bonds between the rings of saccharides. For your proteins, just know that the amine group on the end of the protein has 2 specific ends. Those are going to be covalent peptide bonds formed between the amine group and the carboxyl group on the ends of the amino acid. They have a directionality. That means they have to form from one to the other. All right, what else do you need to know about protein structures? You need to know the 4 levels of protein folding. The first is just the sequence of amino acids, and that's just those links in the chain. Amino acid 1, 2, 3, 4, whatever. That's your primary structure. Your secondary structure is the folding of that chain into things called alpha helices and beta sheets. And that happens due to hydrogen bonds between those functional groups that are attached to the amino acid. The tertiary structure, that's when the protein starts getting its 3D shape, and that happens from non-covalent interactions between different R group atoms. That takes away free energy, so the protein becomes much more stable. 4th degree is when multiple polypeptide units link together. The best example that we have in this class is hemoglobin, making that sort of 4-leaf clover structure out of 4 different tertiary units of hemoglobin protein. What else do you need to know about proteins? Proteins can be denatured either through extreme chemical environments or extremely high temperatures. And when that happens, those linkages between all those R groups start breaking down and the protein will no longer do its biological function. It's basically molecular machinery that won't work anymore because we have chemical bonds breaking down. All right, I put the captions back up because this does get a little bit information dense. Let's talk about nucleic acids. Nucleic acids are linear sequences of nucleotides. And we talked about what a nucleotide was. It's that 5-sugar ring, the phosphate group, and the nitrogenous base. Now, nucleotides have a 3 and 5 direction, and that just depends on where on the 5-sugar ring the phosphate hooks up to. We number that sugar ring, and one of those ends up being number 3, and the other one ends up being number 5. And so there is directionality to nucleic acids. There are 2 types of nucleic acids, and they are DNA and RNA. Let's talk about ways to remember the different types of nitrogenous bases. Okay, some nitrogenous bases have 2 rings, and those are your purines. All right, so take a look at your purines up there. One way to remember purines are pure silver. Now, what's the chemical symbol for silver? It's Ag. And adenine and guanine start with A and G. I always like to think of silver rings in a wedding, and you need 2 rings usually at a wedding. So those have 2 rings. So to remember that, purine, pure silver, you can hear the word pure there, and you need 2 rings to get married. So double rings, pure silver, those are your double ringed nitrogenous bases. Your pyrimidines down there, those are single rings. And I always think of a single ring like a pie. Pyrimidine is 1 ring. So like the pie goes all the way around and makes 1 ring. Now, when you look at these, just remember that we always have A pairing with T and G pairing with C. All right, this is our final topic. Thank you for sticking with me through this whole thing. And remember, this is a whole unit, so it's coming at you pretty fast, and I hope you'll go back and watch it multiple times. In this unit, we're going to be describing the structural similarities between DNA and RNA. All right, let's go ahead and talk about the structure of nucleotides one more time. We still have that 3 components. We have a sugar, a phosphate, and a nitrogenous base. We have them connected by covalent bonds to form this linear molecule with a 5' and 3' ends. And we also have nitrogenous bases that are perpendicular to the sugar phosphate backbone. So that just means the lines, like the ladder rungs, are at a 90-degree angle to the phosphate background, which is the blue strip here versus the colorful bars that are going across the page or left to right. So your phosphate backbone is going up and down and twisted, and your nitrogenous bases are going side to side. All right, if you want to look at the specific differences between the 2 sugars, we have deoxyribose over here in DNA, and look at the sort of bottom of the ring in the front. You've got OH and H, that's deoxyribose for DNA. And over in RNA, there's 2 OH hydroxyl groups on the bottom of that ribose sugar. So that's a very subtle difference between those 2 sugars, but that's one of the things that makes them different, is one's ribose and one is deoxyribose. The other thing, DNA is almost always double-stranded. RNA is single-stranded. The other big difference is the nucleobases. All of them use the C, G, and A, but in RNA, T is replaced with U. So those are your big differences between DNA and RNA. The other ones are the locations they're found. DNA is usually in the nucleus. RNA is found out in the cytoplasm in multiple forms. You've got rRNA, tRNA, mRNA, which we'll do all those whenever we talk about protein synthesis. All right, I think that wraps up chapter 1. I hope this has been helpful. I'd really like your feedback on how I can make these better for you. I'm not particularly used to teaching by video. So how can I make them better? Do you like the captions down at the bottom? There were some scenes I put those on and some scenes I didn't put them on. I notice with my hillbilly accent, they don't always get what I'm saying exactly right. So let me know if they're good enough or if they're kind of, don't put those on there, Mr. Sater. All right, so that's Unit 1. All the pictures I put on this video should be on the page where you found this video. I'll assign some FRQs for you to practice this Unit 1 material.